Hello again, everyone. I am Shahram Kordasti from King's College London with my colleague Arjan van der Loosdrecht from Netherlands. We are going to sharing this session on immunology and microenvironment in MDS. Welcome, and we are going to start with the first presentation by Professor Marc Raaijmakers from Netherlands. Marc, over to you. And the title is Niche Immunology Taxonomy in MDS.
Thank you, Shahram, and thank you, Arjan. And thanks all for having the opportunity to present and share some data with you. The original title was rather nondescript about taxonomies and immunology. So I'm going to share with you some new data that are at the interface between the bone marrow microenvironment and the immune microenvironment and its role in evolution in lower risk MDS. There is no relevant disclosures. So very briefly as an introduction, so there is insight mostly from mouse models that primary alterations not per se in hematopoietic cells, but in stromal microenvironmental cells can lead to leukemia predisposition and ultimately a clonal myeloid disorder. And this again is mouse models that have shown this, and typically in the context of MDS, AML, but also CMML. But coming from these mouse models, I think there is a number of questions that should be asked, and these are the questions that my lab and other labs are working with. So first of all, is there a necessary contribution of the bone marrow stromal niche to MDS initiation and evolution? If so, how does that work? Is an aberrant environment a driver of mutagenesis, or is it more a selector of clones? What would be the molecular determinants of such processes? And last but definitely not least, what's the, if any, relevance to human disease? Because most of the data that I showed you was in mouse models. So to address these questions, we have invested in generating a taxonomy of the human, normal, and leukemic marrow, where we can really look at the signaling between hematopoietic stem cells and progenitor cells and their stromal and endothelial niches. And we did that by an approach where we purified these cells and then subjected them to single-cell sequencing. And this approach we have now also used in lower-risk MDS. So what you see here is data from the normal bone marrow. These are 5 donors of normal bone marrow and low-risk MDS. This is composite data of 5 low-risk MDS patients at diagnosis. These were mostly SF3B1 and or TET2 mutated. And what you can appreciate is that using this approach, there is a robust representation of stromal cells in these taxonomies. Typically, they are rare in our aspirates, but with this pipeline, we have robust representation in these taxonomies. And they are characterized by specific markers like the leptin receptor, CXCL12, and KIT ligand. And the latter two, as you know, being key HSC, HSPC supportive factors. So here we have this data. And then first, if we look at the population level of stromal cells, and then at their transcriptome, comparing normal bone marrow to low-risk MDS, I think the most dominant aberrant pathway is inflammatory signaling. In particular, NF-kappa beta mediated inflammatory signaling. And this is, I think, consistent with earlier data, and this is at the protein level, looking at phosphorylated P65, where you can see that in the lower picture, in low-risk MDS, there is NF-kappa beta activation in stromal cells, but also in hematopoietic elements, and I'll come back to that. Now, what if you zoom in and dissect this at a single cell level within this stromal population? That is shown here, and basically to summarize this, what we observe, and this is not unlike what we observed in AML, is remodeling of the stromal compartment with a relative loss of stromal cells that have high expression of KIT ligand and CXCL12. And there is remodeling towards an inflammatory population in orange here in lower risk MDS. And this inflammatory signaling again is TNF-alpha signaling via NF-kappa beta, at least according to the annotation of gene expression. Now, is this then common in low risk MDS, or is this restricted? Again, this is only data of 5 patients. So what we did there is calculate a score that reflects the amount of inflammation in stromal cells by looking at the differentially expressed genes in this inflammatory cluster. And by doing that, by generating such a score, you can now go to a larger cohort of low-risk MDS patients. And these are patients from the OVAN 89 study. And we looked at 45 patients where we did bulk RNA sequencing on the stromal population and then calculated the amount of inflammation in stromal cells. And you can see in this graph that if you compare that to normal bone marrow, this is a rather common phenomenon in low-risk MDS, although it's variable. The amount of inflammation in stromal cells varies, but again, typically higher than in the normal marrow. Is this related to the genetic background? Well, not very clearly, at least not in this cohort that we studied. So this is looking at the amount of inflammation in stromal cells. Yellow is high inflammation, blue is low inflammation, and you can see that there is not a clear pattern or clear association with the genetic background. Now, another question is, and Shashi already this morning mentioned that this is a very relevant question in the field, what causes this inflammation? So with single cell data, you can approach that at least to some extent, looking at predicted signaling. And when we asked the data, where would predicted TNF-alpha signaling occur between which cell types, what the algorithm told us is that we may want to look at interactions between bone marrow stromal cells and CD8 T cells. And why is that? That is because the stromal cells have the highest expression of the canonical receptors for TNF-alpha and interferon gamma of all the cells in the human marrow, which is, I think, quite interesting. And then if you look at where in low-risk MDS are TNF-alpha and interferon gamma expressed, it was in a subset of CD8 T cells. And here we provisionally annotated as a CD8 effector memory interferon gamma high subset of CD8 T cells. This is all predictive. Is there any anatomical substrate to this? So then you can look at the proximity of CD8 T cells and stromal cells in the low-risk MDS marrow. And this is 2 representative pictures showing you that there is an intimate anatomical relationship between these cells. Based on this data, we estimate that about 20 to 30% of CD8 T cells in the MDS marrow are very closely connected to stromal cells, here identified by CD271 as a marker. Well, what about the physiology? Can T cells inflame stromal cells? Here we took an initial approach, which is an ex vivo approach, where we isolated T cells, we activated them, took the supernatant and exposed stromal cells to that supernatant. And what you can see in the left lower graphs is that this resulted in a transcriptional profile that is indicative of NF-kappa beta activation, including downregulation of KIT ligand. And this was very reminiscent of what we observed in our low-risk MDS patients. And this was partially, at least, dependent on expression of the TNF-alpha receptor. If we knocked that down, this inflammatory activation was mitigated. So this tells us that T cells, at least in this setting, could play a role in the inflammation of stromal cells. So I don't think that's the only pathway. I think there is good data to suggest that downstream progeny of a myeloid clone may also play a role in inflammation of other cell types, including stromal cells. But still, this leaves unanswered the most important question: what is the relevance or the consequence of stromal inflammation? How does that affect normal hematopoietic stem cells versus mutated hematopoietic stem cells? Now, in order to address that, we first looked at the same 45 samples at the gene expression in hematopoietic stem progenitor cells, but now separating the cohort that had high inflammation in stromal cells versus low inflammation. And what this data shows is that if there is high inflammation in stromal cells, you also have activation of inflammatory pathways and inflammatory stress, TP53 pathways in your hematopoietic cells. Now, this is an association in human low-risk MDS. Is this a causal relationship? What's the chicken, what's the egg? In other words, can the inflamed stroma induce these abnormalities? And how does that affect function? So that's why what we did here. Here we switched to the mouse system, where we isolate stem progenitor cells from mice and then co-culture them on stroma in which we genetically induced NF-kappa beta activation. And we did this by constitutive overexpression of the IKK. And what we then observed is that if you take normal hematopoietic stem cells, co-culture them with this inflamed stroma, you see again activation of these inflammatory pathways, at least at the transcriptional level. Again, very reminiscent of what we observed in low-risk MDS. If you look more specifically and ask the question, how much of these changes in this experimental system do we actually see in human disease? We concluded that about 15% of the differentially expressed genes in hematopoietic stem progenitor cells in low-risk MDS, in comparison to human normal marrow, is actually induced by this system, which may indicate that if we look at differentially expressed transcriptomes in low-risk MDS, a considerable part of that transcriptome may be inflammation-driven or even stromal inflammation-driven. And this overlap we dubbed, and I'll come back to that, stromal inflammation-induced gene alterations in HSPCs. And again, if you look at these genes, they are implicated again in apoptosis, MAPK signaling, TNF signaling, inflammatory signaling, also viral infection, which may be interesting in the context of that T cell link. Now, how does this affect function? If we took these HSPCs coming from the inflamed stroma, their CFUC capacity was quite dramatically diminished, and also when we then transplanted these cells into mice, they had clearly reduced repopulation ability. So there is functional impact of the inflamed stroma. Then, of course, what about mutated cells? Because these are non-mutated HSPCs. First, looking at the gene expression again in our single-cell data. So here we identified within the single-cell taxonomies mutated cells, in this case SF3B1 mutated cells, by several approaches, 10x, RAID-seq, and also computational modeling, splice-up, showing that they are mostly in the HSPCs, but for example in the NK cells you can also see that there is a mutated fraction, and this has been described before, but not in stromal cells and also largely not in T cells. But then if you look at the mutated cells and compare them to the non-mutated cells, you can see that these inflammatory signatures are mostly there in the non-mutated cells. while the mutated cells relatively express signatures that would be indicative of cell survival and proliferation, suggesting that mutated cells may be relatively resistant to this inflammatory signaling. Again, we put that into an experimental setting, here using again the ex vivo IKK overexpressing stromal cells. And then what we observed is that this reduces the number of normal non-mutated HSPCs, also the number of SF3B1 mutated cells. These were from patients with a 50% approximately VAF, so mostly SF3B1 mutated 34 positive cells, but you can see to a less extent. So this, although I would consider this preliminary data because it needs to be repeated, may be indicative and in line with the notion that mutated cells may be relatively resistant to that suppressive effect of stromal inflammation. What about the clinical relevance? Looking at patients with a high inflammation in the stroma versus low inflammation. Patients with high inflammation do worse regarding overall survival. Then this is looking at stromal cells. If you now look again at the transcriptome of hematopoietic stem progenitor cells, but using that site signature and then selecting genes that are of prognostic value, we can see that that inflammatory profile in hematopoietic cells is of course related to stromal inflammation and therefore also significantly related to outcome in this cohort. But this hematopoietic signature allows us to look at other independent cohorts, and this is one of them, confirming that inflammation in hematopoietic cells that can be induced by stromal cells is related to outcome, and in this cohort quite strongly to leukemic progression. So where does that leave us? And this is my last slide. This is a model we proposed a number of years ago, but I think data would still be consistent with that, that in MDS there is occurrence of mutations that may be stochastically or not, but that these mutated cells lead to an inflammatory environment that may be direct or via myeloid progeny or via innate adaptive immunity, and that inflamed stroma then leads to an inflammatory environment that represses normal hematopoietic cells, non-mutated, but mutated cells may be relatively resistant, relatively expand, and perhaps, and this remains to be seen, in an inflammatory environment that may or may not be mutagenic, have the ability to build more mutations and ultimately progress towards leukemia. Again, still I think a working model, but it's something that we like to build on in our work. So with that, I'll stop thanking the people who did the work, mostly Lampeng Chen, a postdoc, and Yuji Bian, a PhD student in my lab, and it would not have been possible through collaborations with the people that are listed on this slide. Thank you.
Okay, thank you very much, Marc, for excellent talk as always. We just have time for one question because Job.
So Marc, great talk. Over here. I'm not going to argue with you about the role of stromal cells because I'm going to lose that battle, but do you think that there is also still room for direct effects of the mutated MDS cells directly without going through the stromal cells in suppressing normal hematopoietic cells? Do you think that the mediators that you've identified can also explain some direct effects?
Yeah, I think that's very well possible, Joop. I think there is at least two things to consider here. You have to define inflammation, and you have to define the molecules that you consider inflammatory molecules, and you have to look at their ability to act to cells that are further away. There is some cytokines like DAMPs that really, I think, work at cellular resolution. And then there's others like perhaps IL-6 that have the ability to have a wider range, let's say. And that brings you back to the anatomic context. And I think their stromal HSPC niches may be particularly relevant, although I think they're unlikely to be the only one. And the other thing is that a bit of outside of the context, but indirectly related to your question, we also have to think about inflammatory impressions or epigenetic changes that endure, and then if that would be in a stromal compartment that we think is relatively robust with a relatively low turnover that may remain there, that could also be relevant if we think about recurrence of disease and later effects. So short answer, no, I don't think stromal cells are the only thing. I think other cells can do this, but I do think that there is a rationale to at least focus on stromal cells among other cells. Thank you for the question.
Okay. Thank you very much, Mark, for the great talk.
Then we continue with Daniel Starczynowski from the US, from Cincinnati Children's Hospital, and he will talk about dysregulated innate immune signaling in MDS, in line with Mark's talk, maybe.
Very much so. Thank you for the introduction. Thank you to the MDS Foundation for hosting another fabulous event. It's a really pleasure and honor to be here. So I have a long-standing interest in understanding the cellular, molecular, and genetic basis of MDS, with a particular interest in how inflammation and innate immune signaling contributes to the pathogenesis of these complex diseases. And as Marc just so elegantly pointed out at the very end of his talk, not all inflammation is the same. We have to define the inflammatory signals because they have very unique and particular functions on the MDS clonal population. So I'm going to tell you today one story. It's a recent story that just came out a couple of weeks ago, how we identified a signal from our gut that contributes to the expansion of pre-leukemic clones. No relevant disclosures. So as you've heard this morning, mutations, somatic mutations in our hematopoietic stem cells, particularly involving genes of DMT3A, TET2, and ASXL1, contribute to clonal hematopoiesis. And this is an age-related phenomenon that results in expansion of these pre-leukemic cells. And I think this could be the next major health crisis, and I'll tell you in a moment why. Clonal hematopoiesis is a precursor to clonal cytopenias of undetermined significance, or CCUS, as well as MDS. At least in the United States, greater than 20% conservatively of 70-plus-year-olds have CHIP or CCUS. So that represents, just in the United States, over 10 million individuals. So why is it potentially a next major health crisis? Well, first of all, CCUS results in persistent and untreatable cytopenias. Individuals with CHIP or CCUS have an increased risk of hematologic malignancies, and that risk is about a 1% annual increased risk. But that's probably not the real cause we should be concerned. It's really the increase in all-cause mortality and increase in cardiovascular disease that we need to be worried about. So although CHIP has an expansion of these mutant cells, these individuals are largely asymptomatic. But these mutant clones do promote inflammatory and immune-related diseases, which are thought to be contributing to this all-cause mortality. We also know that chemotherapy and different types of inflammation can select for these mutant hematopoietic stem cells. And the question is really how. And in some cases, we have understanding, but particularly in the context of inflammation, we still don't have a really concise understanding of why these mutant cells respond in the way that they do. And through that understanding, are we able to target these pre-leukemic states using novel therapeutic strategies? So how can we prevent CHIP or CCUS? First of all, who's at greatest risk? The strongest predictor of CHIP and CCUS-associated health conditions is the actual size of that mutant clonal population. So if you have a small pool of cells and it remains small, then you're largely going to be fine. But if that pool of cells, mutant cells, continues to expand, you have this increased risk of not only heme malignancies, but these cardiovascular diseases. So it's really not clear why some individuals with clonal hematopoiesis have an expanding pool of clones. Moreover, therapies targeting CHIP-associated mutations, such as in DMT3A, TET2, ASXL1, really don't exist. They're not very effective. And general anti-inflammatory therapies have shown limited benefit. So if you dial down that inflammation through general strategies of tuning down inflammation, those have largely not worked because they're too non-specific. And so the goal for this project that we had was to identify an age-related factor that contributes to the expansion of these mutant pre-leukemic clones. And if we can understand that mechanistic basis of why these cells continue to expand, perhaps we can intervene at the most earliest stage and prevent that expansion. So the question really started about a potential link between our gut health and pre-leukemia. There were several studies showing an association with inflammatory bowel diseases and expansion of pre-leukemic cells. And so Puneet Agarwal, who led this study, asked a simple question: is there a relationship between our gut health and pre-leukemic cell expansion? And so just as a brief reminder, when we're healthy and young, our intestinal epithelial lining is intact, our microbiome is rich in diversity. But as we age, that intestinal health starts to dissipate. We have disruptions of the intestinal epithelial lining. The integrity becomes compromised. You can start getting leakage of material from the lumen into the circulation. This is also true in individuals, younger individuals post-chemotherapy. That's very abrasive to the epithelial lining. And in the most severe cases, as I just pointed out, inflammatory bowel disease has complete loss or terrible loss of the intestinal epithelial integrity. And so the idea is that there's increased risk of pre-leukemic cell expansion as we have loss of that intestinal epithelial integrity. So we decided to address this question, and before I jump into the experimental design, I just want to point out, I'm going to show you several of these schematics demonstrating bone marrow transplants. We use DMT3 mutant cells to study pre-leukemic cell expansion, but we had to modify our transplant conditions in these mice, and so we couldn't use high-dose radiation because it's incredibly disruptive to the gut and the microbiome. So we identified an ultra-low radiation conditioning using 2.5 Gray, where we maintain the gut health and we don't change the microbiome. So all the transplants I'm going to show you use this low dose bone marrow conditioning. And so in the first set of experiments, we asked if we use recipients that are either young or old, do these DMT3 mutant cells expand? And it's consistent with other studies. The DMT3 mutant cells do not expand in healthy mice, but they do in an aged mouse recipient. And you can see the expansion of the hematopoietic stem cell compartment as well in the bone marrow of these mice. We can do the same experiment now taking young mice, right, where we don't see expansion of these DMT3 mutant cells. We can damage their gut using dextrin sulfate sodium, which mimics an inflammatory bowel disease. And so if we disrupt the gut, you can see this profound expansion of the DMT3 mutant cells in the stem cells in the bone marrow, as well as this clonal expansion in the periphery, as you can see here on your right side. So loss of gut integrity induces expansion of the DMT3 mutant cells. So loss of gut integrity, as I mentioned, as we age, results in dysbiosis in the microbiome as well. So we wanted to ask whether changes in the microbiome are contributing to the expansion of these pre-leukemic cells. So we did several experiments, the first of which is we did fecal transplants into recipient mice. And so we used fecal material from young mice that had disrupted microbiome, again using DSS as a conditioning regimen. And so if we transplanted the DMT3 mutant cells and introduced fecal material, an unhealthy microbiome, now we can see expansion of these mutant hematopoietic stem cells in the bone marrow. In a similar line of experiments, we took fecal material, aged microbiome from older mice or younger mice. And again, it was only the microbiome through the fecal transplants that expanded the DMT3 mutant cells originating from the old mice, not the young mice. So it's changes not only in the gut epithelial integrity, but also changes associated with the microbiome that contribute to the expansion of these pre-leukemic cells. So we wanted to examine the microbiome in a little bit more detail, so we performed 16S RNA, ribosomal RNA sequencing. And here's some of the results that are summarized in terms of the different microbes that we see in the mice. And we have 4 groups here just to orient you. We have healthy young mice, young mice that are treated with DSS, again, to disrupt the gut as a positive control, and then young healthy mice and old mice. And I'm not going to go into the details and specifics of the microbial changes, but I think what you can appreciate, as in these old mice, you see a lot more red and orange bars, which represent various gram-negative bacteria, as compared to young healthy mice, which you see a lot more gram-positive bacteria. We see a similar phenomenon in humans, where, again, young donors have a lot more gram-negative bacteria, a healthy microbiome. But as we age, or in MDS, or in the extreme case of IBD, you see a lot more gram-negative bacteria expanding in the gut, but also remnants in the circulation. So loss of that gut integrity results in dysbiosis, changes in the microbiome that represent an increase of gram-negative bacteria. So we wanted to understand whether this increase in gram-negative bacteria might be contributing to the expansion of these cells, and here we used an antibiotic treatment experiment to diminish the amount of either gram-positive or gram-negative bacteria. And so we used a variety of antibiotic cocktails, as shown here. So V represents vancomycin, which preferentially targets gram-positive. MGN is a cocktail of 3 antibiotics that preferentially targets gram-negative bacteria. And so when we eliminated the gram-negative bacteria in these DMT3A mutant chimeric mice, we saw suppression of those pre-leukemic cells. But we didn't see suppression of these leukemic cells when we eliminated the gram-positive bacteria. So this suggests that gram-negative bacteria are contributing to the expansion of these pre-leukemic cells. And so now the question is, mechanistically, how? So one of the key distinguishing features between gram-positive and gram-negative bacteria is this outer membrane of gram-negative bacteria, which contains lipopolysaccharide, which creates the structural integrity to the bacteria. LPS on its own is actually immunogenic. It actually has variable immunogenicity when it activates its receptors, such as the toll-like receptors on the mammalian cells. But interesting, LPS can also be not only immunogenic, but non-immunogenic in certain cases, and actually dial down inflammation. So we don't think just the presence of LPS is contributing to this phenomenon we were observing with pre-leukemic cell expansion. And so a few years ago, there's a seminal study revealing that an intermediate of the lipopolysaccharide pathway that generates part of the inner core through this pentose phosphate pathway in bacteria generates this highly immunogenic byproduct called ADP heptose. ADP heptose is, again, as I mentioned, a byproduct of LPS generation, but it's released during not only the growth, but also lysis of gram-negative bacteria and can freely penetrate mammalian cells. So we asked whether ADP heptose, this metabolic byproduct of gram-negative bacteria, is elevated in the circulation of our older mice as well as in humans with MDS and CHIP. And for the what we used is mass spec to first measure the circulating ADP heptose. So you can see here in the plasma and also in the bone marrow, you see in mouse models of gut injury, here we just injured the gut with DSS, and you can see this expansion and increase of ADP heptose in the circulation. And in healthy mice, when we don't damage their gut, there's essentially no detectable ADP heptose in the circulation. If we do the same sort of experiments in the plasma from healthy donors, either young or old, you can see as in the older healthy individuals, there's already circulating ADP heptose. We see a similar phenomenon in MDS in CHIP individuals, and as well as in younger individuals, much younger than the MDS cohort, with that inflammatory bowel disease, and you can see this elevated ADP heptose. So ADP heptose does indeed circulate upon loss of gut integrity and expansion of the gram-negative bacteria. So the fundamental question is ADP heptose sufficient to induce the expansion of these pre-leukemic cells? And the answer is yes. So in the top there, I have the DMT3A model again transplanted and grafted into those younger mice. We have a low chimerism, but if we introduce ADP heptose through the circulation, through a tail vein injection, you can see the expansion of the DMT3A mutant cells in the bone marrow, and this coincides with a competitive fitness advantage in the periphery as well of these mutant cells. And again, as I showed you earlier, DMT3 mutant cells that don't see ADP heptos do not expand in this model. Does this extend beyond DMT3 mutations? And the answer is yes. We examined other models of DMT3, including knock-in mutation, as well as TET2 mutations, and we see a similar phenomenon that ADP heptos in a chimeric model of TET2 mutant bone marrow induces expansion of the TET2 mutant stem cells, as well as increases our competitive fitness advantage in the peripheral blood. And lastly, we saw that ADP heptos also expands primary human MDS34 positive cells in vivo. I guess compared to just water-treated MDS cells, these cells don't expand, as has been demonstrated many times. But if these cells see ADP heptos in circulation, we see the expansion of these MDS cells in these xenografted mice. So ADP heptos is one of the key mechanisms, that byproduct from the gram-negative bacteria that drives the expansion of these pre-leukemic cells. So how does ADP heptos drive the expansion of these cells? What is the molecular basis for this? So ADP heptos is recognized by what's thought to be a single mammalian kinase receptor, and it's an atypical kinase called ALPK1. This was shown by a seminal study a few years ago by Zhao et al. ALPK1, this receptor for ADP heptos, is overexpressed in an activated state in human MDS and AML stem progenitor cells. And in other work, ALPK1 activating mutations associated with these rare solid tumors, as well as these inflammatory conditions called ROSA syndromes. So I'm just going to kind of walk you through some of the mechanisms. So ADP heptos in the circulation can freely penetrate the mammalian membrane. And in the cytoplasm is the receptor, ALPK1, which is this atypical kinase. And once ADP heptos is recognized by ALPK1 kinase, it becomes allosterically activated. It then phosphorylates this little protein called TIFA that's in the cytoplasm as well in a dispersed distribution. When TIFA is phosphorylated by ALPK1, it forms this signaling complex called a TIFAsome, which recruits TRAF6 and other signaling intermediates that can activate NF-kappa B signaling. So we found that ALPK1 and TIFA are both overexpressed in pre-leukemic cells, including DMT3, TET2, among others, and as well as in human MDS CD34 positive cells. So you can see here the levels of ALPK1 protein expression, as well as TIFA expression, compared to healthy 34 positive cells is much higher. And if we take these cells and treat them ex vivo with ADP heptos, you can see how sensitive and responsive they are to NF-kappa B activation as compared to healthy cells. And so ADP heptos is recognized by ALPK1, forms a signaling complex called TIFAsomes, specifically in these pre-leukemic cells. So we did the essential genetic rescue experiment where we eliminated ALPK1 from the DMT3A mutant pre-leukemic cells and found now that these cells are completely unresponsive to ADP heptos-induced expansion. You can see that the complete loss of the stem cell expansion capacity, not only in the bone marrow, but also in terms of their competitive fitness advantage. So ALPK1 is essential for these pre-leukemic cells to sense and respond to circulating ADP heptos. So we wanted to extend our observations and examine additional cohorts of healthy individuals for ADP heptos. And for this, we established a reporter system called a TIFAsome assay, where we simply added a fluorescent marker, TD tomato, to TIFA, and we introduced those into a human AML cell line, THP1 cells. And these reporter cells can be activated and monitored for TIFAsome formation. And we wanted to use this reporter system to add plasma onto these cells and ask, is there sufficient levels of ADP heptos to induce TIFAsome formation in these cells? So in the first experimental setup, we just took these reporter cells and added ADP heptos. So without ADP heptos, TIFA, these TIFAsomes, are not formed. You can see the diffuse expression of TIFA in the cytoplasm. But as soon as they are sensing ADP heptos, and we just add very low quantities of ADP heptos to these reporter cells, you get these puncta that form, and this is an indication of TIFAsome formation. So if we use this assay and now sprinkle on plasma from young individuals, you can see there's no TIFA-zone formation. However, plasma from healthy older individuals has plenty of ADP heptos in circulation to induce these TIFA zones. And the same is true for MDS and CHIP patients. So it's quite striking that even low amounts of ADP heptos in circulation, the plasma and the bone marrow can induce these TIFA zone formations in leukemic cells. So we used this assay, scaled it up using image stream, and so we monitored not only additional mouse cohorts, but also human samples. But just for proof of principle, older mice have elevated ADP heptos in the circulation and sufficient amounts to induce TIFA zone formation, not only in the peripheral blood, but also in the bone marrow. And then here's the TIFA-zone assay, again, to infer the levels of ADP heptos in the circulation. And you can see young individuals essentially have undetectable amounts of ADP heptos. There's insufficient amount, if there is any, to induce TIFA-zone formation. Again, healthy individuals that are older have sort of a dichotomous expression pattern of ADP heptos, whether either you don't have any or you do. And we see a similar phenomenon in MDS and CHIP. Particularly in CHIP, if you look, some CHIP individuals do have a mutant cell population, but they don't have ADP heptos while others do. So we wanted to look at this cohort a little bit more carefully and ask, if you have CHIP, so you have a detectable pre-leukemic cell, and you're positive for ADP heptos, are you any different than if you have CHIP, but the absence of ADP heptos? And the answer is yes. You have an increased risk of some cardiovascular abnormalities, including hypertension and venous thromboembolisms. So although this is a small cohort of CHIP individuals, if you do have a pre-leukemic cell and you have circulating ADP heptos, you have some of the associated conditions that we're so worried about in individuals with CHIP, such as cardiovascular disease. If you want to learn more about this TIFA-zone assay, and we'd be happy to run your samples, just follow the link there. So this is my last slide. I just want to summarize some of the key points. First off, as we age, we have compromised gut health. This is just a normal part of aging, and that's associated with dysbiosis of the microbiome, increased expansion of gram-negative bacteria, and circulating ADP heptos, this byproduct of gram-negative, healthy or pathogenic gram-negative, it doesn't really matter. And ADP heptos induces a competitive advantage of the mutant cells when it reaches these cells, and it activates ALPK1 in the pre-leukemic cells. This induces TIFA-zone formation. And what I didn't show you, it induces a particular set of NF-kappa B related genes. So these cells are very sensitive to ADP heptostimulation and induces a set of transcriptional programs that allow these cells to self-renew and proliferate rapidly, and is very distinct from other inflammatory mediators such as IL-1, LPS, TNF-alpha. We also show that if you block the signaling axis, if we intervene and block ALPK1 signaling in these pre-leukemic cells, we can prevent the expansion. So the take-home message is that there's an age-related metabolite that's in our circulation, ADP heptos. It's a dichotomous factor. If you're young, you typically don't have it. As you age, the levels increase, and this drives the expansion of these pre-leukemic cells. And so, as I mentioned, this paper was just released, so if you want to take a closer look, please follow that link. And then just like to acknowledge Puneet Agarwal, who's a senior scientist who drove this project, and Avery Sampson in the lab who developed the TIFA-zone assay. And also acknowledge collaborators and funding sources. So thank you very much.
Daniel, thank you for this excellent talk. And the floor is open for a couple of questions. Please go ahead.
Wonderful talk, Dan. Just a question about where do you think that the ADP heptos is coming from in aging individuals? Yeah. So, I mean, the TIFA-zone assay shows that there's ADP heptos, but they don't have IBD. Do you think it's just slow breakdown of the gut? Or do you think there's other sources?
Yeah, no, it's a great question. We really only address the association with the gut, so we think it's just that natural aging, loss of gut epithelial integrity that happens, and those slight changes is probably enough to induce, result in the circulation of ADP heptos from those gram-negative bacteria that are expanding. We've also found that gram-negative bacteria not related to sepsis is in our circulation, so healthy gram-negative bacteria And even those could be a source of ADP heptos as well. So, but there's certainly other sources. There's mucosal and other sources of gram-negative bacteria that we haven't looked at that could also be contributing. But it's a really important question. Thank you.
Valeria?
Thank you, Daniel. Always wonderful data. Very, very challenging and interesting. My question is, you showed that, in fact, ADP heptos is much higher in inflammatory bowel disease than in CHIP, than in even in MDS.
Yeah.
How many of the inflammatory bowel disease patients do have CHIP? And should we worry about that and do intervene in that cases?
Yeah. It's a great question. One thing before I answer your question, I want to make one specific comment. And so I don't think ADP heptos is resulting in mutant cells. I think there's two unfortunate events. Yeah. So there's two unfortunate events. You have pre-leukemic cells and you happen to get a little bit older. And so to answer your question is, I don't remember the specific frequency of IBD having CHIP. It's not very high, but it was enough to pique our interest. But I'm not sure that we have to necessarily be worried about it, but it certainly helped us frame a hypothesis to test. But I don't want to leave you thinking that we have to start checking IBD patients for CHIP.
Thank you. And a final short question.
Yazan from UT Southwestern. Thank you for a fantastic talk. My question was actually very similar to Dr. Santini's. And to frame it in a different way, for patients older than 60 or 70 with IBD, I mean, those we would anticipate, if they have pre-existing CHIP, should be at much higher rate of transformation. And I just wonder if your comments and thoughts about that.
Yeah, so I want to be careful with the description of transformation. I think maybe clonal expansion.
Sure.
Certainly checking, I don't know how often IBD patients are checked for CHIP, but certainly that has to be interrogated. But I think I'd rather not maybe focus so much on IBD, but things that disrupt our gut and the integrity of our gut when we're younger. Most obvious for this group is various forms of chemo and targeted therapy, right? That happens as part of the treatment regimen. What that's doing to the gut obviously is profound. What it's doing to ADP heptos, we're only just starting to scratch the surface, but I think that just serves as an exemplar of how we should be thinking about what ADP heptos could be doing when you disrupt the gut, which we, for the most part, haven't paid a lot of attention to.
Thank you.
Finally, where is the innate cell? What is it? Is no monocyte or whatever the cell might be in between?
Sorry, I missed the first part.
Is there no innate immune cell, as we know, in between the pathophysiology you described, between the gut and...
So, really good question. So there are normal immune cells that are associated with the gut lining that normally sense ADP heptos, but we don't think it's those cells that are contributing in any way, at least in our model system, to the expansion of these pre-leukemic cells. So it's really the pre-leukemic cells that have expressed ALPK1 from 0 to quite a bit that are sensing it. Now they have the receptor to sense that circulating ADP heptos.
Clear. Thank you, Daniel. We move on to the final of this first part.
The next speaker, Professor Amit Verma from Albert Einstein College of Medicine in the US, and he's going to talk about targeting inflammation in MDS.
Thank you very much. Thanks for the invitation. I wanted to show two relatively unpublished stories on trying to target two distinct inflammatory mediators in translational studies in MDS. And these are my disclosures. Okay, so the idea of targeting inflammation is not new. And I think a lot of people sitting in this audience have done seminal work showing that inflammation is important and a driver. And what I'm going to focus on in the first part of this talk is the effect of inflammation on the anemia phenotype. Now, all of us know that anemia is the predominant clinical manifestation phenotype in MDS, and even low-risk disease in MDS, where anemia is the main phenotype, as we all know, the survival is not so good. So if we can figure out ways to improve erythropoiesis I think we'll do a huge service to our patients. So we tried to target therapeutically a well-known mediator of inflammation, which is a protein complex, a heterodimer between S100A8 and S100A9. These are proteins that have been in multiple publications, a lot of people in this audience have shown are elevated in MDS models, and they are very potent drivers of myeloid-derived suppressor cells. These are cells that are myeloid in origin, not fully differentiated. They secrete a lot of suppressive cytokines and result in immunosuppressive microenvironment. So this is just a simple cartoon. S100A8A9 heterodimers bind to a set of cognate receptors. These are 3 that have been well studied in myeloid cells: toll-like receptor 4, RAGE, CD33, and the downstream signals, what I show for TLR4, is actually through the midosome and IRAC, and this is been well-studied pathway that's overactivated in MDS. This is a study that we did, which was led by Dan, which showed that IRAC is actually primed and overactivated. So anything that signals through TLR4 is already primed to be overactivated. And at the end, NF-kappa B is a driver of these signals, leading to more perpetuation of inflammation. S100, this is just a snapshot of a bunch of very important studies that have shown that these heterodimers are elevated. This is data from Marc's study, where he showed that mesenchymal cells in MDS can also secrete this heterodimer, and it's prognostic. And this is the data that we mined using Dr. Boltzwood's CD34 data set. We looked at expression of all toll-like receptors, and TLR4 was about 3 times overexpressed when you look at normal CD34s. And it did show overexpression, not just for TLR4, but for its cofactor, which is known as LY96 or MD2. In the same data set, we also looked at S100A8 and A9 and tried to do clinical correlations, and found that when MDS patients samples have high levels of A8 and A9, they tend to have worse hemoglobin, so more severe anemia. It's not just a player in MDS, but in myelofibrosis also. Dr. Schneider's work and other people have shown that the fibrotic phenotype that's seen in MPNs in myelofibrosis are also driven by this heterodimer. So we started an antibody campaign to find specific monoclonal antibodies that could block the heterodimer. We inoculated a higher mammal to find more potent antibodies, a long drawn-out process. And in the end, we have been able to generate a pipeline of potent monoclonal neutralizing antibodies. And this is data showing that these monoclonals can inhibit the binding of the heterodimer, not just to TLR4 and RAGE and 33, but also we have found some other receptors that are important in the inflammation cascade, and also CD69, which is not just on myeloid cells, but a prominent receptor on lymphoid cells. When we treated primary MDS samples, bone marrow derived, in the presence of these monoclonals, we did see an increase in erythroid colony formation. As you can see, the top panel shows control isotype antibodies, and the lower panel is treatment with one specific monoclonal against the heterodimer. Across a set of different mutant subtypes, we do see an increase in CD71 and glycophorin A positive cells. We have now humanized these rabbit-derived antibodies. This is data, part of the data that we show, 2 AML samples that were derived from MDS, and we saw increase in vitro erythropoiesis in these AML samples also in the presence of the humanized neutralizing antibody. So we hope we can get this antibody into the clinic relatively soon, and I'll be happy to give you an update. The second part of the talk, I wanted to show you some data with an inhibitor of the inflammasome. Now, inflammasome, again, is a very important mediator of disease in MDS, and the NLRP3 inflammasome, in particular, has been shown to lead to conversion of pro-IL-18 and pro-IL-1 beta to their active isoforms, and also result in gas-dermin decleavage, caspase activation, in the end causing MDS-like phenotypes. So when we looked at these inflammasome components in the same MDS CD34-derived data set, we did find overexpression of these downstream inflammasome mediators in MDS. Also, some of these mediators were prognostic. So high IL-18, high gas-dermin D levels were predictive of poor survival and also predictive of an increased risk of AML transformation in this MDS transcriptomic data set. The inhibitor that we then used was a slightly unconventional way of inhibiting NLRP3. So there is a protein, NEX7, that is an essential adapter for the assembly of the NLRP3 inflammasome. And a company, Halia Therapeutics, developed an allosteric small molecule inhibitor that binds to NEX7 and disables the assembly of NEX7 into the NLRP3 inflammasome. This inhibitor locks NLRP3 in its inactive stage and has been shown to be safe in human volunteers, and we expect the human trial to open in low-risk MDS hopefully this year. So we've generated a lot of data with this particular drug, HT6184. It can decrease IL-1 beta secretion in myeloid cells. It can suppress the production of IL-6, and we show in IP experiments that it actually binds very potently to NEX7. When we performed colony assays with primary patient samples and this particular drug, we again saw increase in erythropoiesis and decrease in the myeloid bias. And here you see some samples with all different kinds of mutations. Mechanistically, it has been shown that activation of NLRP3 inflammasome and subsequent activation of caspase can actually result in cleavage of the master transcription factor that drives erythropoiesis, GATA1. So we looked at GATA1 expression, and we can see that the presence of this NEX7 inhibitor does increase total GATA1 levels. To show the relevance of NEX7 in the activity of this drug, we have CRISPRed out NEX7, and when we CRISPR out NEX7, we don't see this activity of this drug in a cell line model, showing that NEX7 inhibition is important for the small molecules activity. Finally, we've done a bunch of in vivo studies. This is PDX in NSG mice done with a high-risk MDS patient in pre-treatment. No real difference, but 4 weeks of treatment with this inflammasome inhibitor resulted in significant decrease in the MDS clone in these NSG mice. So the company has actually done a small trial in India, and they have not released the full results. It's expected to be sometime later this year, but what they have released is what I'm showing you, that this study has achieved their expected endpoint to continue with a second larger cohort. And this is just example of one transfusion-dependent MDS patient who was single agent treated with this particular drug. So we hope to see this drug in action in a broader study later this year. So just to summarize, the two pathways that we've focused on in our lab, S100, A8A9 heterodimers, also known as alarmins, DAMPs, calprotectin, with a specific monoclonal antibody-based approach, and then targeting the NEK7 NLRP3 inflammasome with a small molecule. The idea is not just to increase red blood cells and decrease transfusion dependence, but also to target, the crippling fatigue and the impaired quality of life by targeting inflammation, which most of us know that, in our patients, even with transfusions, even with drugs that reduce transfusion dependence, the fatigue still does not go down as much as we want. So we feel that targeting inflammation may help us achieve that. Rangbao in our lab led the S100 studies. Rahul and Divij led the inflammasome studies. And this is just a very short list of giants in the field of MDS that we have had the pleasure of collaborating and have supported us. And these are the two companies that are now trying to develop these drugs for clinical use. Thank you.
Thank you very much, Dr. Verma, for the wonderful talk. I may start with my own question first. When we talk about controlling inflammation, obviously we have inflammation in all, most of these MDS in different pathways, but different data, for instance, the canakinumab trial data that published show that, for instance, SF3B1 mutated patients are not responding, and also seems like it's a bit genomic specific. Do you think, first of all, have you seen the same thing with the pathway you are blocking? And also, do you think in future trial of next phase, we need to be more genomic specific?
Yeah, it's a great question. I think that probably will be best sorted out in the first human trial, where if we see a signal. The inherent limitations of ex vivo studies and certainly the animal models for our disease. We have not yet seen an overriding signal of a particular mutation that relies on the inflammasome or calprotectin, but the numbers are relatively small.
Yeah. Okay. Yes.
Yes. Hi, Amit. Wonderful and impressive. My question, you may be aware of the fact that erythropoietin also has several effects, anti-inflammatory, decreases IL-1, decreases IL-6, TNF factor, and I guess that some of your patients definitely were treated with erythropoietin. And my question is, could you separate between the EPO effect and the antibody effect, and maybe what do you think for the future? We should aim for combinations or things like that.
Yeah, no, it's a great question. A lot of our patients were already resistant to EPO and had high baseline EPO levels. I think when these things move into the clinic and move early on, we'll see those effects. Interestingly, there was a paper in leukemia that was published many years ago that showed that alarmins can actually decrease EPO formation by liver cells. So there's quite a complex interplay with all of these cytokines.
Thank you. And the final quick question. Thank you.
From Karolinska. I wonder about the prognostic information that can be derived from inflammation status. Is it an independent factor, and is it relevant both in lower risk and high risk MDS?
Yeah, there are, it's a great question. I just showed the KM curves from Marc's study, which was actually S100A89 derived from mesenchymal cells. There have been similar studies looking at different mediators. There is a study from Dr. Ifantis where he looked at this consolidated inflammatory score, even though it was more of an AML-centric study. My belief is that lower risk also, it will play a prognostic role.
Thank you, Amit. We move on with the selected oral abstracts in this session, and we are happy to ask Aditi Shastri to come to the floor, and she's also winner of one of the Young Investigators Award, as you also had Pedro Mauro this morning. So we are very happy that you are here, and you will speak about environmental pollution triggers inflammation in vivo and is associated with higher risk MDS in real-world urban cohort. The floor is yours.
Thank you so much, and thank you for accepting our abstract for an oral presentation. I'm truly delighted to be here on behalf of all my colleagues to present this work to you, and in a beautiful city in Rotterdam. So to move on. Okay. So worsening air quality is something that, unfortunately, it is a global problem. Whether you are anywhere in Asia or you are in the United States, these are just some recent examples that I found online that I wanted to show you. Or you're traveling, sightseeing. This is something we encounter more and more globally. And as we have actually seen in this session, inflammation is something that is very important, is greatly upregulated in MDS, and we had our wonderful speakers talk about many different aspects of this pathway. However, one of the things that has inspired me to really dig deep more into the epidemiology of why there is increased inflammation, potentially why patients get hematologic cancers, is really in my clinic. This is a question I get a lot, is why did I get MDS, or why do I have a high-risk CHIP or whatever? And unfortunately, we never have a good answer to this question. So we decided to dig deeper into this issue. Of course, there is this multifactorial causation that we know that causes this progression of clonal hematopoiesis to overt malignancies due to this immune selection. And there is selective pressure, which is induced on immune selection by various therapies. Aging is a strong stimulus, and smoking. However, there are environmental factors at play yet too, but they are yet unknown. So this is something that we are very interested in looking into. So the first real clue that there could be some environmental exposures that lead to pre-malignant conditions came from the work of Dr. Rachel Zieg-Owens and collaborators at my institution at Mount Sinai, which showed that the precursor condition to multiple myeloma, called monoclonal gammopathy of undetermined significance, or MGUS, was actually upregulated in the FDNY firefighters that were exposed to the World Trade Center tragedy. And a larger cohort of the FDNY that was also exposed compared to a control population actually had a higher elevation in MGUS, which you can see here in the graph was overall MGUS, the M spike, as well as the light chain, even after controlling for various covariates such as sex, age, BMI, smoking status, etc. And I think some of this data, along with other data, put in place programs for early detection and surveillance for people that were exposed to the World Trade Center dust. There was another important study that was done by the previous speaker, Dr. Amit Verma, also at my institution, which was really looking at the early detection of clonal hematopoiesis in the first responders. of 9/11. And in this paper that was actually published in 2022, we did see that there was actually a much higher rate, higher burden of clonal hematopoiesis across every decade of life in those that were exposed to the World Trade Center versus those that were a control population. And some in vivo modeling of this work actually did show that compared to vehicle or treated mice, the World Trade Center dust-exposed mice actually had more genomic deletions, more indels. They had more synonymous high-impact mutations, as you can see here in this circus plot, compared to controls. A follow-up work that was actually presented at ASH last year actually also shed light on the frequency and pattern of mutations that we can see in the World Trade Center exposed firefighters, with them really exhibiting a very distinct mutational spectrum. They are younger, and this is actually very different from what you see in age-associated clonal hematopoiesis. And there is elevated risk of developing leukemia. And further mechanistic insights via in vivo models really did show that there was accelerated TET2-mediated CH, increased IL-1RAP expression, and mitigating these effects with anti-inflammatory therapies, was something that identified that this is an important possible epidemiologic reason for CH development and progression. So to really look at this in a more, look at it more in a day-to-day life, just look at air quality in general, we decided to actually just focus on the neighborhood where we get most of our patient population from, which is the Bronx. And in the Bronx, New York, we have a fairly large population of 1.4 million people. We have a huge area that we service, which is 25 zip codes, 286 census tracts. And this is actually, unfortunately, the poorest and unhealthiest county in New York State. And we do have a large burden of cancer. And what makes it interesting and unique to study is that the Bronx was a historically redlined area. And what that means is redlining, as some of you in the audience know, was a faulty mortgage appraisal practice in the 1920s, where certain areas in different cities around the world were actually allocated less resources, and the effects of which persist until today. So in these areas that were redlined, you can actually see a higher heat vulnerability index till this date, even though the populations that used to live here have changed over the decades. And today in the Bronx, predominantly the populations that live in the Bronx are Black and Hispanic communities. At the time of redlining, it was actually Italian and Jewish communities that used to live here. And we actually know a lot about pollution's role in malignancy, but we really don't know much in hematologic malignancies. So the first clue to us looking at this kind of genomic work was we also saw in a study that was published not too long ago that exposure to PM2.5 can actually increase the EGFR clonal burden in non-small cell lung cancer across multiple data sets that were studied in different countries. So the first study we decided to do, just to model whether we can actually look at air quality, was looking at multiple myeloma. We actually used 1,600 patients data from the year 1997 to 2018, and we identified about 1,500 patients as having a first cancer diagnosis of multiple myeloma, and about 14 of these patients had addresses available for analysis. And this is actually the cohort that we had. They were 50% Black, 30% Hispanic. Here in this population, and we could see that a large percentage were never smokers, actually, 62%. And I'm proud to say, actually, we have a Netherlands connection, is that we actually collaborate with the University of Utrecht to really use their satellite-derived air quality data, which we're able to actually overlap with our patient data using a statistical model called a land use regression model that has been previously published and validated. And some interesting correlates that we saw of this work was that the patients in our study were actually exposed to, for almost a decade, to the PM2.5 level that was well above the EPA standard. And there was actually a hazard ratio for overall survival for PM2.5 and black carbon that trended towards significance, but was not significant. And I will show you some limitations and how we are overcoming this, but I want to switch quickly to the reason why we're here, which is MDS. And so we actually looked at a smaller cohort of patients with MDS with a very similar race and ethnicity background at our institution. And what we saw was very interesting is that here the black carbon, as if you can see in figure A, correlated with a higher IPSS score for patients with MDS. There were some other correlates too with nitrogenous compounds, etc. The genomic mutations, I'm sorry, I know it's hard to read, but it still works in progress. We could see that individual genomic mutations also had correlates across the pollution rasters with various different pollutants. Across a few different years, as you can see in figure B, C, and E, we actually saw that the IPSS score actually did correlate significantly with black carbon and PM2.5 levels. However, we have looked at these for individual years, including survival, and aggregating the data, it sometimes trends, it has trended to not be significant for survival. So this is some nice data that was given to us to correlate what we're finding from the UK Biobank by Sid Jaiswal at Stanford. And here actually for CH, we could actually see that we had a significant p-value as a correlate for PM2.5 exposure. And this was actually primarily driven by the non-DNMT3A type of CH as well as loss of Y. In figure C, this is actually just shows you the forest plot of the hazard ratio for exposure to PM2.5 and incident MDS, which is significant. And B is really the Kaplan-Meier plot for the cumulative incidence of MDS stratified by the 90th percentile of exposure for PM2.5. So this is actually significant. When we try to dig deeper with in vivo models to really try and understand what is happening at a more granular level biologically, one of the things that we could actually see is in the panel A, this is actually the levels of pollutant chemicals as measured by a chemical analysis, which were higher in samples that we studied in the Bronx compared to controls. And when we modeled exposure in vivo to mice, mouse macrophages, as you can see in B, had higher black carbon compared to controls. And this actually triggers a robust inflammation in the mice with figure C showing IL-1 beta and plasma histamine levels being elevated. And this exposure to black carbon also significantly alters stem and progenitor cell growths and can stimulate the growth of mutant clones in vivo. I know it's a busy slide, I want to point out a couple of things. So here, actually, this is after exposure for 4 months. This is after exposure for 8 months. But across the board, you can see it does trigger a kind of bone marrow failure phenotype that we can see. Here, this is actually a CH model where we used a TET2 mutant CD45.2 cells. And here, actually, after exposure to black carbon, at about 16 weeks, we could see clonal expansion. And in these mice, we also did colony assays, and with methocult, and the number of colonies, as we can see, is reduced. And qualitatively also, we see reduced colonies compared to controls. So there are, of course, some limitations of the epidemiologic and in vivo work. A lot of the work is retrospective. Race ethnicity is self-reported. Migration or occupational hazards is something that's hard to account for. However, in our study, there was less than a 10% migration rate. during the time that we did this study. And of course, survival in MDS is very strongly linked to many other factors, including treatment. And so for future directions, we are testing our hypotheses in murine models, and this is actually, there is a lot of interest currently from active military, deployed personnel in potentially protection or chemoprotection for cancer interception by targeting inflammatory targets of interest. So to summarize, the natural progression of pre-leukemia to leukemia is poorly understood in the context of environmental associations. Studies incorporating cancer epidemiology and mechanistic in vivo studies do give us the best possible opportunities to better understand this. Air quality in our study was above the EPA standards during most of the years we studied. Higher PM2.5 exposure correlated with more CH, more cumulative incidence of MDS in the UK Biobank, and black carbon and nitrogenous compounds are associated with higher IPSS in MDS. In vivo modeling shows more inflammation and reduced healthy erythroid and myeloid colonies upon exposure. There is increased clonality in a TET2 mutant CH model. So with this, I really want to thank everybody in my lab that worked very hard on this, especially three trainees, Megha, Matthew, and Michael, our biostatistician, Kith Pradhan, environmental epidemiology collaborators, Dean Hosgood, George Downward, and Yiyu Zhao, Amit Verma. Thank you very much, and look forward to your questions.
Thank you. We are running out of time, and we have an online meeting next to you, so we are not able to question. Sorry, Pierre, maybe in the pause. We run a little bit out of time. Sorry. Thank you.
Thank you.
All right, thank you very much. The next selected abstract is Willemijn Hobo from Netherlands, going to talk about bone marrow T cells of patients with MDS, display increased effector memory differentiation with high checkpoints expression and retain polyclonal functionality. Very long, sorry. Over to you.
Okay, thank you for the invitation. So the previous talks introduced the impact of inflammation on MDS development, but now I would like to switch gears to the actual immune cells. We know that Myelodysplastic Syndrome is an immunogenic disease, so depending of course on the type of MDS and the risk category, patients can be treated with lenalidomide, HMA, chemotherapy. But high-risk patients can be treated with an allogeneic stem cell transplant, and we know that this therapy can be curative due to the graft versus tumor effect mediated by the donor T cells and the donor NK cells. So we know it's an immunogenic disease, but we want to understand better why those T cells and NK cells don't attack the tumor cells sufficiently. And of course, knowing this, you can think of identifying targets for development of novel immunotherapies. So just a short recap of how the anti-tumor immune responses work. Of course, we have the important immune effector cells, the T helper cells and the cytotoxic T cells, which can recognize, of course, the leukemia cells. Upon activation by an antigen-presenting cells, those T cells can become activated and exert their functions. And within the CD4 T helper cells, we have different subsets where the regulatory T cells and the T helper cells can really favor leukemia development and expansion, while the T helper 1 cells and the cytotoxic T cells can really eliminate the leukemia cells. And of course, those CD8 T cells, upon activation, they clonally expand, produce effector molecules, and can really lyse the tumor cells. On the other end, we have the innate immune cells, the NK cells, another important immune effector population. And again, NK cells can also be activated by dendritic cells, favoring the maturation of those NK cells, which can subsequently attack and kill the leukemia cells. But what about immune responses in MDS? A lot of studies have already been performed. It has been described that the dendritic cells and the monocytes, so the antigen-presenting cells, which are required, of course, for good T cell and NK cell activation, are part of the MDS clone and that it can impact their function. So we have seen in multiple publications that in high-risk MDS, for example, increased abundance of regulatory T cells is observed. In lower-risk MDS, increased abundance of T helper, 17 subsets is seen. For the cytotoxic T cells, it has been described that they have a more exhausted-like phenotype. And on the other hand, for the NK cells, more immature NK cells with less cytolytic capacity have been seen. And this all together, and of course it has also been addressed in one of the previous talks, MDSCs play an important role in dampening T and NK cell responses in MDS. And this all together can of course also contribute to the development and progression of MDS. But those are still all mostly individual studies where different subsets or phenotypes were looked at, and we wanted to do a more integrated, comprehensive analysis. So actually we analyzed all different immune subsets in MDS bone marrow, but today I will only focus on the T cells. Our patient cohort consisted of 14 age-matched healthy donor bone marrow samples, 13 lower risk MDS, 14 intermediate risk, and 19 higher risk samples. And we always, at each analysis day, we included a reference sample for potential normalization. We applied high-dimensional flow cytometry and clustering analysis with over 20 parameters. Well, you can imagine, when you do manual analysis, you can do many different combinations you can look at. So we really need computational tools to look to the features of those T cells and the differences between controls and MDS patients. So we applied dimensionality reduction analysis and also flow sum clustering analysis. But in order to do such kind of extensive analysis, you need a very good workflow. It all starts with good preparation, with the panel design, choosing the titers, quality control, machine standardization. Of course, then you continue with the analysis, compensating, gating, exporting populations of interest, then further sample selection, all kind of other quality controls, so transformation, downsampling, and then eventually you can do the clustering and dimensionality reduction analysis. And finally, you can also still apply some extra visualization steps. So going to the data, we first looked to lymphocyte composition in MDS patients. So these data are from, well, the C stands for control, low risk, intermediate risk, and higher risk MDS patients. And we look to the frequencies of T cells, B cells, and NK cells within the bone marrow mononuclear cells. And with increasing MDS risk score, we saw a reduction in number of T cells and also B cells, while NK cells were still quite stable. And looking to the relative contribution, again, you see that there is a slight increase, although here it's not significant in the abundance of the T cells. And interestingly, within those T cells, we observed that the CD4-CD8 ratio was reduced upon increasing MDS risk score. So higher risk MDS patients really had a lower CD4-CD8 ratio, meaning increased abundance of CD8 T cells as compared to CD4 T cells. And this is also depicted here in another way where we look to the percentage of blasts in the bone marrow. And also regulatory T cells within the CD4 T cell subset, we saw an increase in the presence of those regulatory T cells with increasing MDS risk score. Then we wanted to know more about the T cell activation and differentiation state of those T cells. So like I said, dendritic cells present antigens to naive T cells. If the T cell harbors the respective T cell receptor, the T cell becomes activated, and of course, it also needs signaling activation. And then upon activation, the T cell can differentiate to all those different types, so stem cell memory T cells, central memory, effector memory, and eventually effector T cells. Within the MDS patients, we observed that there was a strong skewing within the memory T cell differentiation. So here you see on the left for the CD4 T cells, you see in the controls and in the higher risk, those are the naive T cells. And you see that there are much less naive T cells and much more effector memory T cells within higher risk MDS. And it's even more pronounced in the CD8 T cell compartment. And that's also depicted here. So there's a strong skewing towards terminal differentiation of the T cells in MDS. But of course, it's also dependent on the actual functionality and the phenotype of the T cells. And we know that checkpoint molecules are very important for T cell activation. And too much activation, so regulation by the checkpoint inhibitors, too much activation of T cells leads to autoimmunity, while too much dampening of T cell functionality via checkpoint inhibitors can support tumor growth. Looking to those checkpoint molecules, we observed that within higher risk MDS patients, multiple of those molecules were higher expressed: PD-1, TIGIT, KLRG1, CD27. And this is also depicted in another way. Within higher risk MDS patients, there's increased co-expression and increased expression of those checkpoint molecules. But we know that also the expression of those molecules differs with the differentiation stage of the T cells. So here we really looked within the different T cell subsets, memory subsets, to their contribution to the expression of the respective molecules. And indeed, the increased expression of those checkpoint molecules could partly be related to the increased skewing to effector memory differentiation. But nevertheless, also when we do computational analysis, we see reduced abundance of the more immature, naive-like T cell subsets, and we see increased abundance of the more terminal differentiated T cell subsets with high expression of the different checkpoint molecules in higher risk MDS. Then we performed functionality assays because, of course, checkpoint expression can be a signature of T cell activation, but it can also be, of course, a feature of T cell exhaustion. So that's why we performed functional experiments. We used the bone marrow mononuclear cells, let them rest, then we activated the T cells overnight with PMA ionomycin, or for 4 hours with PMA ionomycin, and then we looked at all different kinds of T cell effector functions. And interestingly, here I immediately focus on the memory T cell subsets to correct for potential impact of the skewed differentiation. Looking within the memory T cells of controls, low risk, intermediate risk, and high risk MDS patients, we actually observed very effective production of interferon gamma, IL-2, TNF-alpha, and also degranulation marker CD107A. And also the proliferation capacity of those T cells was still retained. So it's really interesting. They have an increased skewing towards terminal differentiation, high expression of checkpoint molecules, but still they are capable of producing cytokines and proliferating. And very shortly, for the CD4 T helper cells, we observed similar patterns. So also increased expression of checkpoint molecules, partly related to their memory differentiation state, but still reduced abundance of the more early memory T cells or naive T cells, and more increased abundance of the effector memory T cells with high expression of the checkpoint molecules. And also within the CD4 T cells, we observed that those T cells had retained functionality, or even here within the memory T cells, we observed actually some increased interferon gamma production, which might maybe be interesting in the context of the inflammation, which was previously addressed, and also retained proliferative capacity. So to summarize, We observed a decreased CD4-CD8 ratio in higher-risk MDS patients, increased memory differentiation with high expression of checkpoint molecules, which could partly be related to the differentiation state, though with retained proliferative capacity and cytokine production capacity in response to polyclonal stimulation. So the T cells still have a highly activated and differentiated phenotype, but with retained polyfunctionality. And I think this also still warrants further studies. This is at diagnosis or in an early stage, but what happens in time? What are the T cell features, the dynamics, the functionality over time of those T cells, and how does it contribute to MDS pathogenesis? Because we know with MDS progression, the leukemic cells escape and the immune system does not respond. So yeah, we want to further address this in follow-up studies to eventually identify new targets for adjuvant immunotherapy. And finally, I would like to thank all the people involved, especially Dide, who performed all this work. And of course, this work was also done in collaboration with Amsterdam, and I would like to thank you for your attention.
All right. Thank you. Thank you very much for the very interesting talk. Unfortunately, because of the time, we don't have time for question. We need to move immediately to the next session. And after that, we have a guided poster session to remind everyone. I would like to thank all the speakers for beautiful talks, and thanks for listening. And we move without any break to the next session.